Okay, so at this point we have done the first part, the second part of the test review. So we're going to take those, we're going to take them, get rid of them, and we are going to fold this over, and then I'm going to fold this over like that, so that the only bit we're looking at are these parts. Okay, and bring this down a little bit like that, so we are good to go. Okay, so these are some old favorites and some things that you're just reviewing, because remember, when you take the STAR test, it's not just going to be on that unit, it's going to be on stuff that you have done previously. So looking at this, let me just straighten that up a little bit. Okay, so I have 4 organelles here. This first one, just by looking at it, you should know that this is a mitochondrion. mitochondrion, mitochondrion. And what do mitochondria do? I know you should all by now be shouting at your video saying they do cellular respiration. They take glucose and oxygen and they convert it into carbon dioxide and water and ATP, which is what the cell uses for its to drive all its processes. This one here, maybe not one of my best diagrams. This is a ribosome, and there is literally one thing that we associate with ribosomes. This is where messenger RNA is used as a template to assemble amino acids in a particular sequence, aka translation, and that produces proteins. So ribosomes and protein synthesis go together. Okay, this next one, all right, a little more unusual. Maybe we haven't talked so much about this one recently, but this here is a lysosome. Lysosomes are the cell's waste disposal. They break down materials. Lysosomes can build up materials as well, because lysosomes contain enzymes. And enzymes, as you know, are proteins which speed up the rate of chemical reaction by reducing the activation energy required for that reaction to happen. And if I say grandma's pizza, you should remember what enzymes do. This last one, probably one of the more familiar organelles. This is the Golgi body. And what does the Golgi body do? Well, after ribosomes have helped make proteins, the Golgi bodies, they modify them, they add bits to them, they fold them into a specific shape, and they package the proteins and send it off to where it needs to get to. So modification and packaging of proteins. That's the Golgi body. Okay, while we're talking about cells and organelles, I guess we need to talk about prokaryote and eukaryote again. Okay, you should have these down cold by now. The number one question you need to know is, how do you distinguish between a prokaryote and a eukaryote cell? In other words, what are the differences? How are they different? How are they the same? Well, they're both cells. But you know that prokaryotes, first off, the only type of living organism that has prokaryotes is bacteria. So all other stuff, plants, animals, protists, fungi, all of those things, they are eukaryote cells. The other thing is that prokaryotes do not have organelles, do not have membrane-bound organelles. You know, all the stuff that you see inside the cell, eukaryotes do. They have organelles. Okay? That says organelles. And this is the big one. In prokaryotes, the nucleus is absent. No nucleus. In eukaryotes, the nucleus or the nuclear membrane is present. So, big way to distinguish between a prokaryote and a eukaryote: the presence or absence of the nucleus or a nuclear membrane. If it's a bacteria, it's a prokaryote. If it's any other type of living organism, it's a eukaryote. Okay, I put this here, and again, this is something we have talked quite a lot about in this unit and in another one, in the previous one. Gametes and somatic cells. Gametes, obviously, sex cells. Somatic cells are body cells. In any organism, there is only one type of gamete. So in male chickens, there are sperm cells. In male sunflowers, there are pollen cells. In female chickens, there are egg cells. In female sunflowers, the female part of the sunflower has the eggs as well. So gametes, sex cells, somatic cells, body cells. Remember, gametes are N, which means that they have single chromosomes. Somatic cells are always 2N, which means their chromosomes are in pairs. Okay, so looking at all these cute little organisms here. So we have, I don't know, a goldfish, okay? And we have a pretty pink flower, okay? And a person and a bacteria, okay? So the question is, what do they have in common? Okay, and the one thing that we know is that all living things, what do you notice? They all have DNA. So that's DNA, that's DNA, right? This is DNA, and this is DNA. And guess what? Those DNAs are all made up of nucleotides. Phosphate, deoxyribose sugar, and a nitrogen base. And so in this one, they have ATGC. This one has ATGC. This one has ADGC. And this one has ATGC. So they all have the same nitrogen bases. And those nitrogen bases do the same complementary base pairing, A with T, G with C. And when they open up and we do protein synthesis, that DNA codes for mRNA, and the mRNA, 3 bases on mRNA is a codon, and we use the codon chart, and that tells us which amino acid it is. The codon chart you would use for all these things is exactly the same, and the reason for that is we have what's called the universal genetic code, okay, which means that in all living things, they all have the same nucleotides. That is why you can take DNA that codes for insulin in a human, put it into the bacteria, and the bacteria will use its protein making systems to make human insulin. The reason for that is that we have the same nucleotides, the same ATGCs, and they all code for the same amino acids. Speaking of which, in this next section here, this should be pretty straightforward. This is all about protein synthesis, and the only thing that I do not have on my review here is I do not have my codon chart, which I'm searching through here to get hold of, and it is, let me go, there we go. All right, the codon chart. You will need, I'm going to scoot this up here because we will need the codon chart here in a minute. Okay, because this was what we did in the previous unit, and it's still kind of important that you remember how to do it. So I have a sequence of DNA. Step number 1, you remember this, break it down into 3s, right? And I need to transcribe the DNA into messenger RNA. I need to do transcription. So if I have an A here, remember mRNA does not have T, it has a U instead. So I have a U. If I have a G, it's going to be a C. If it's a T, it's going to be an A. Then a C is going to be a G. A G is going to be a C. And an A is going to be a U. T will be an A will be a U, C will be a G. Okay, so I've done that section. The next part, I'm going to go ahead and I'm going to break this into 3s so I do not mess this up. If my codon, my mRNA is UCG, remember I go to the codon chart, first letter is U. So everything across here is U. Second letter is C. Everything down here, the second letter is C. Third letter is A. Come over here to third letter, and that will code for SER, serine. G, C, U, G, C, that's in this box, and U is up there. So I'm going to write A, L, A. And then here, A, U, G, A, U, and G, that is going to be methionine, MET. Okay, so break it into 3s, complementary base pair. Remember that instead of a T, you're going to write a U, and then use your codon chart. And again, I'm going to reiterate this bit. If you see a question in a test that has a codon chart in it, you have to use the codon chart in order to be able to answer the question.